Okay, folks. Whoa. Where's my voice? Hello. There we go. Look what I have. Now, before you come up, all right, here's the rules. Here's the rules. You get one card. You have to get it yourself. You can't get it for your friend, and you have to get it right here for me. So come up and get it. Okay, it looks like we have juice. So, if you hear that acting up again, would you do me a favor again? And yes, thank you. All right. All right, so I told everybody in class the rules on the cards. I'll post those rules on the webpage since the camera ran out of juice, and so everybody who wasn't here will see the rules for the cards. That'll be on the class schedule page. So, we don't have a lot of material to cover, which is why I'm not too worried about missing some time today. But I do have, obviously, things that we need to talk about. So this is the second lecture on glycogen metabolism. And last time I introduced the topic by talking about how it is that glycogen is broken down. And the breakdown of glycogen is very simple. You've essentially seen it. Okay? You have a glycogen phosphorylase, which cuts glucose 1-phosphate units off using a phosphate to within 4 units of a branch. Excuse me. You have a phosphoglycerate that converts the glucose 1-phosphate into glucose 6-phosphate so it can be used in glycolysis or gluconeogenesis. And then that branched molecule is dealt with by an enzyme called debranching enzyme that takes 3 of those glucoses and puts it on the other end, making 1,4 bonds, so there's no branch, and then clips that last glucose off using water so that you have free glucose. And at that point, what you have is a molecule that looks like what you see on the screen above, which is the free glucose on top of it, and the, in this case, linear chain, or a lengthened linear chain of the glycogen below. Now, that linear chain has 1,4 bonds, not 1,6 bonds. Okay? And so we have now put this out there available for glycogen phosphorylase to come along and do its thing. Okay? Everybody understand that? Okay. So, moving along then, what we need to do is talk about glycogen synthesis. So that's all there is to break down. All right? We could talk about it basically in 3 enzymes: glycogen phosphorylase, phosphoglycomutase, and debranching enzyme. We're going to see a similar thing with glycogen synthesis. All right? Glycogen synthesis goes through a little bit, one different thing. It's not just the reverse of the breakdown, but you'll see it's not at all complicated. Glycogen synthesis actually starts with glucose 1-phosphate. How would I get glucose 1-phosphate if I didn't have any glucose 1-phosphate? It's a good exam question. How would I get glucose 1-phosphate if I didn't have any? What's that? Galactose would break down and give me glucose and you say, did you say galactose or lactose? So galactose, no. Galactose would not give me that. Not directly, anyway. What's the simplest way for me to get glucose 1-phosphate? Yes? What's that? Can you use a kinase? Well, specifically what? What would hexokinase give me? Glucose 6-phosphate. How would I get glucose 1-phosphate? Phosphoglycomutase, right? So I could go from glucose to glucose 1-phosphate in 2 steps. I could go glucose to glucose 6-phosphate with hexokinase, and then phosphoglycomutase to give me the glucose 1-phosphate. So there's a variety of ways that I could get glucose 1-phosphate to make glycogen with. Okay. Glucose 1-phosphate, however, cannot be added directly to a growing glycogen chain. And there's a reason for that. It takes energy. And you know it takes energy as a result of something I talked about last time. What was that? What was the evidence last time that there was energy that needed to go in to make that bond? Back there. You got energy when you broke it. Very good. What was the evidence I got energy when I broke it? I didn't make ATP. No. What was the evidence that I got energy when I broke it? Didn't get any DH. No. What's that? Nope, not heat. Back there. Didn't require ATP to put the phosphate on. Extra credit point, okay? Didn't require ATP to put that phosphate on there, remember? That's a phosphorylasis reaction. To put that phosphate on there takes energy, and the best evidence that that bond has energy is it doesn't take any ATP to put the phosphate on. Okay. Now, that's important. So if we're going to make a bond that has a lot of energy, we've got to make something that's got high energy to put into creating that bond. And that's what's happening right here. We create something that I call an activated intermediate. And activated intermediates turn out to be really important in biochemistry. An activated intermediate, I'm going to give you a definition here. An activated intermediate is a molecule that has a high energy bond. It's a molecule that has a high energy bond, and it uses the energy of that bond to donate a part of itself to something else. So an activated intermediate is a molecule that has a high energy bond, and it uses the energy of that bond to donate a part of itself to something else. The activated intermediate that we use to make glycogen is known as UDP glucose. Okay? And UDP glucose is shown on the screen. And the high energy bond occurs, all right there where those phosphates are. Right there where those phosphates are. The bond between the glucose and the phosphate is a high energy bond. That bond will be broken, and glucose will be added to a growing glycogen chain in the next reaction I'm going to show you. So UDP glucose is, in fact, a high energy bond. The enzyme catalyzing that has a mouthful of a name. It's called UDP glucose pyrophosphorylase, and it's an enzyme that I think you should know. Nobody likes that name. So I usually give the class a chance to call it what they want to call it. We have to agree on a name. What would you like to call this enzyme? Yes? Larry? You can name it anything you want to name it. We have to agree on what it's going to be. What will we name it? Larry's one nomination. Are there other nominations from the floor? Stephen. We got Larry and Stephen. Yes? Kevin. Oh, I don't want to call it Kevin. Yes. What? Dan. Okay. Larry, Stephen, Kevin, Dan. I'm going to take one more name. So, what's that? UDGP. Okay. That's not really, I'll disqualify that one. That's not really a name. That's an abbreviation. So, all right, what do you got? Wilson. Romney? I still didn't hear what he said. Waldo? Oh, okay, Roldo. Gary. All right. I'm going to pick out about 4 or 5 of those, and we're going to vote on one, okay? We don't take too long with this. All right. First, I had Larry. How many for Larry? How many for Stephen? Stephen's not a very popular name. How many for Gary? How many for, what was another one I got? I still don't know what that name is. Oh, Harambin. Harambin, okay, Harambin. So far it's between Larry and Harambin. Dan, there was a Dan, I think, also. Dan? Between Larry and Harambin, are we in consensus? All right. How many for Larry? How many for Harambin? I think it's Larry, guys. All right. We'll just keep this from the TAs, okay? We'll just keep this from the TAs. So we're going to call this enzyme Larry. It's interesting that name has come up before, and the class has also voted for Larry before. I'm not quite sure why that's the case, but all right. All right. Moving on in our discussion, we come to the next step, which involves putting that glucose onto the growing glycogen chain. This involves important enzyme glycogen synthase, and as we will see, glycogen synthase is very important for the regulation of glycogen metabolism. There are only two enzymes that have roles in glycogen metabolism. One is glycogen synthase. The other is the one I've already talked about, which is glycogen phosphorylase. Okay? Now, I spend the rest of the time talking today after I finish the synthesis to start talking about that regulation, and I'll finish that up on Friday. All right? This reaction that you can see on the screen is catalyzed by glycogen synthase, and you can see the addition of the green glycogen. It's on the upper left of the UDP glucose being added to the growing glycogen chain. You see that it is put on in an alpha 1,4 configuration. Alpha 1,4 configuration. Okay? And you've grown glycogen. That's all there is to it. Okay? Really straightforward synthesis, except for it's missing one thing. What am I missing? Is there a question back here? Yes? Well, so far I've talked about glycogen synthase and Larry. Okay? But there's got to be something else. What are we missing here? What's that? Alpha 1,6? Yeah. Where do the branches come from? Well, the branches come from another enzyme that's got a mouthful of a name, but we're going to call it branching enzyme, guys. That's a simple one here. Branching enzyme simply takes a long chain of glycogen with alpha 1,4 bonds and grabs a hunk of them. And no, I don't care if you know how many is a hunk, all right? It grabs a hunk of them and creates an alpha 1,6 linkage. Why doesn't that take any energy? Alpha 1,4 has more energy than alpha 1,6. How do you know alpha 1,4s have more energy than alpha 1,6? Yeah. Because of what? What doesn't take any energy? True, but it could be equal, for example. So how do you know that there's more in an alpha 1,4 than there is in an alpha 1,6? Yes, sir. The what? Well, steric hindrance plays a role, but there's actually some evidence that you've seen that an alpha 1,4 has more energy than an alpha 1,6. What's that? Yeah, when we had to break the 1,6, okay, we had to use water. That meant that there wasn't enough energy to put a phosphate on. So therefore, right, the alpha 1,4, which we used phosphate to break, means it has more energy because it puts the phosphate on. So very good, very good. Okay, extra credit point there. Okay, that makes sense? So that's all it takes to make glycogen. It takes glycogen synthase, it takes Larry, it takes branching enzyme. That's all it takes, as long as we have glucose 1-phosphate. So the synthesis and breakdown of glycogen are the simplest pathways that you're going to learn in biochemistry. They've only got 2 or 3 steps, and that's really all that there is to them. Okay? The regulation of glycogen, however, is more complicated. And it's more complicated for a very important reason. Glycogen is a very essential resource for cells. Cells use glucose for all kinds of energy. It's the best source of rapid energy that cells have because it's soluble in the water of our bloodstream. So it can travel through our body very quickly, and it gets into cells and can be used very quickly. Glucose doesn't have as much energy per carbon as fatty acids do. And you say, well, why don't we use fatty acids? Because fatty acids can get broken down inside of cells. And fatty acids do get used, but fatty acids aren't easily soluble in the liquid environment of our blood and have to be packaged up. We'll talk about that next term. They have to be packaged into something called lipoprotein complexes. Okay? That takes a while. You don't want to be packaging up your complexes when you're trying to escape that grizzly bear, right? So it's important then that we have a rapid energy source, and glucose is that. So because glycogen is the storage form of that glucose, we have to be careful. We don't want to break it down too quickly and lose it all, and we don't want to be making it when we're trying to escape a grizzly bear. So we've got to be able to very quickly turn on and turn off glycogen metabolism, okay? And we're going to see the glycogen metabolism tends to go on or off in terms of making or breaking down. Okay? Because of that, the enzyme that breaks down glycogen, which is the place where I start, has two very important regulatory mechanisms. One that is allosteric and one that is covalent modification. Now, that means that when we look at covalent modification, we can have, in this case, the modification is phosphorylation. We can have a phosphate on or we can have a phosphate off. The form of glycogen with the phosphate off is known as glycogen phosphorylase B. And that's also abbreviated GPB. It's on the upper left of the screen as you see. The form of glycogen that has a phosphate on is known as glycogen phosphorylase A, or GPA. And it's the form that's shown in the upper right. It's also the form that's shown in the lower right. And GPB is also the form that's shown in the lower left. So everything on the left is a GPB. Everything on the right is a GPA. Well, if we go from left to right, that means we're either adding a phosphate or removing a phosphate, depending upon which direction we're going. And if we go up and down, we're talking about allosteric regulation, meaning binding of a small molecule to affect the enzyme. That was the definition of what allosterism is. By the way, that was a question several of you had on the exam that I regraded. The regrades are back in the BB office if you want to pick those up. Okay. All right, so going up and down, all right, we have allosteric modifications. So that means, therefore, that the glycogen phosphorylase B can exist in two different forms. We can have a GPB in the R state, and we can have a GPB in the T state. We can have a GPA in the R state, and we can have a GPA in the T state. Now, this on the surface looks very complicated, and I'll be honest with you, it's a little complicated, but it's not quite as bad as it seems. Let's remember some guiding principles. R state, T state changes always involve allosterism. They don't involve covalent modification. That's true for any enzyme. Allosteric modifications do not involve covalent modification. They involve binding of a molecule. Okay? So as we go up and down, we're binding a molecule. As we go left and right, we're putting on or taking off phosphate. All right. Now I'm going to set up and show you some specific examples. I'm just going to briefly talk you through what's on the screen here. Then I'll show you some specific examples of each of the GPBs and GPAs. Okay? Now, when would we have the phosphorylated form of glycogen phosphorylase? Under what conditions will a phosphate get put onto glycogen phosphorylase? What hormone stimulation will ensure that gets put on? I heard somebody say glucagon. That's correct. What other one? When what? Epinephrine and glucagon. Right. So epinephrine, glucagon stimulation of cells will favor the phosphorylation of a whole series of proteins, resulting in phosphorylation of glycogen phosphorylase. Under what conditions do phosphates get removed? Insulin. Insulin stimulates the activity of phosphoprotein phosphatase, which takes phosphates off. So as we move to the right, that means we've had epinephrine or glucagon stimulation. And as we move to the left, it means the cell has gotten insulin stimulation. Okay? Cells get insulin stimulation when the body is full of glucose, and they're stimulated to take it up and reduce the blood glucose concentrations. Yes? So when you go to the right, that means that the cell has gotten stimulated by epinephrine or glucagon. Moving to the left, stimulation by insulin. Okay. Now, And you can see in the covalent modification there in the center where it says 2 ATPs to 2 ADPs, that it takes 2 ATPs to make that happen. This is a dimer, meaning it's 2 subunit enzyme. Each subunit gets a phosphate. And the phosphate is a little hard to see in there, so it's sort of a blue circle that appears in the upper molecules on the right, I should say. Okay, well, let's look at some specific examples. All right, let's think about what goes on in the liver. Looking here specifically at glycogen phosphorylase A in the liver. This is the easiest one to understand. Okay? Glycogen phosphorylase A is the form of glycogen phosphorylase that people refer to as being, quote, the most active. It's the most active, the most active form. Well, why do they call it the most active form? Because you've actually got two forms. The R state is going to be the most active form, wouldn't you say? Because the R state of enzymes are usually the most active form of enzymes. So how do you have the most active form in the T state? Would that be active? What do you think? Would the T state of glycogen phosphorylase A be very active or not? How many say yes? How many say no? The no's win, and the no's are right. Okay? So why do we call this the most active form? Why do we call glycogen phosphorylase A the most active form if it has an inactive form? Well, you'll see why in just a second. Okay? It relates to the allosteric interactions with the glycogen phosphorylase A. Glycogen phosphorylase A can bind to glucose. Under what conditions is glycogen phosphorylase going to bind to glucose? It's a simple one, guys. When there's plenty of glucose. Is the cell energy state going to be high or low? When the cell has plenty of glucose, the energy state of the cell will be high. Okay? When the energy state of the cell is high, does the liver want to be breaking down glycogen? No, right? So, in that case, the abundance of glucose indicates to the cell that we've got plenty of energy and the enzyme shuts down. It goes into the T state. So why do we say that this is the most active form of the enzyme? Notice that there's nothing on the left side indicating moving up to the R state. There's no allosteric effector that converts the T state into the R state. What does that mean? It means that when there's no glucose, the enzyme automatically goes into the R state. Only when there's glucose will this enzyme get put into the T state. So most of the time, the cell doesn't have a lot of glucose sitting around because glucose is a poison, right? Most of the time, the cell doesn't have a lot of glucose sitting around, and so the enzyme is mostly active because it automatically flips into the R state. Only when there's abundant glucose will the enzyme say, okay, we got to shut down. We don't want to be breaking down glycogen. Does that make sense? Yes, Professor, that makes very good sense. Okay, I like that answer. All right. Question, yeah. Is glucose a substrate? It's not a substrate for the enzyme, no. But glucose is an energy source for the cell. So glucose, the only thing glucose is doing here is it's an allosteric effector. Remember, something that binds to an enzyme and affects its activity. Glucose is binding to the enzyme and converting it into the T state. Remember how ATCase bound to CTP and converted it into the T state? Same thing here. Let's go out to the muscle and see what's happening in the muscle. And let's think here about glycogen phosphorylase B. This is what we call the less active form of glycogen phosphorylase. And you see these terms are a little muddied, aren't they? All right? Because there's also two forms of glycogen phosphorylase B. The two forms of glycogen phosphorylase B are R state and T state. And let's look to see what converts them into each. What converts glycogen phosphorylase B from the R state into the T state? Two things will do it. ATP, glucose 6-phosphate. What do you suppose that indicates? High energy. Under high energy conditions, we don't want to be breaking down our glycogen. We shut off this form of glycogen phosphorylase. Make sense? most of the time, cells have abundant ATP and glucose 6-phosphate. Most of the time, glycogen phosphorylase B is in the T state. This is why we say it's the less active form of glycogen phosphorylase. What turns it on? What puts it into the R state? That's AMP. And AMP, I hope you've learned by now, is an indicator of low energy. When cells have low energy, they want to be breaking down their glycogen, release glucose so they can make energy. Really, really important. Well, how do I know if the cell has glycogen phosphorylase B or glycogen phosphorylase A? Well, if I don't give you any other information, you might say, well, it's got both. If I had both and the enzyme and the cell was in a high energy state, what state would glycogen phosphorylase B and glycogen phosphorylase A be in? They'd both be in the T state, right? I'm sorry. In high energy, they'd both be in the T state. That's right. Okay, excuse me, I got it backwards in my head. They'd both be in the T state because we would have glucose, which converts the A into the T state. We would have ATP and/or G6P, which would convert it into the T state. If we have low energy, what's going to happen? No glucose. GPA is going to flip into the R state. GPB is going to have AMP present, and it's going to flip into the R state as well. You see how these work together? Let's imagine I'm getting ready to run that race, and I go out and I run that race, and my muscles are using glucose faster than I can get it delivered. I'm using ATP faster than the creatine phosphate can convert ADP back to ATP. What's going to happen to my ATP levels? Right? What's going to happen to my AMP levels? Right? Free sound effects, guys. All right? We're going to have AMP going high. The very first thing that's going to happen in those muscle cells is that GPB is going to convert to the R state. I'm not going to hardly have any GPA. Why? Because it takes a blood supply to deliver hormone from the source of where it comes from to the liver to let go of glucose. Yes? Question? Yes? No, cyclic AMP activates protein kinase A. No. So the only way you get cyclic AMP is by adenylate cyclase. Right. So that's it does not convert into cyclic AMP, no. There's no relationship between AMP and cyclic AMP. No. Okay? Now, think about that. So your body is immediately flipping glycogen phosphorylase B, which is sitting there in your muscles, into the R state before the hormone can get there to tell the cell to start making B into A. That's really good because we've now seen a couple of examples of how the muscles are set up to instantaneously provide nutrients and energy when they need it. And our muscles are the things that keep us alive. We have to have those energy needs fixed instantly. Creatine phosphate does that for us. And now we see glycogen phosphorylase does that for us as well. We don't have to wait for the hormone, which might take a few seconds to get there. The muscles keep on cranking it out because they instantaneously do this flip. Everybody understand that? Okay. Well, I've got a song today that is a song I've never sung to a class before. You guys are going to be the guinea pigs for this. It's a very difficult song to sing, and I don't have a recording of it, which means yours truly is going to have to sing it because I doubt anybody in here knows the song either. Yeah. So please bear with me. The title of the song is It Flows, Then My Glycogen Grows, and it's to the tune of an old 70s song called Love Grows Where My Rosemary Goes. Has anybody in here actually ever heard that song? Okay. Maybe you can help me. All right. Sing loud. Here's how it goes. When I'm exercising, it is not surprising. Insulin is absentee. But when it flows, then my glycogen grows, and nobody knows like me. During all the flexing, it may seem perplexing, liver's working easily. Turning lactate into glucose is great, and it is first rate to me. There's something about an insulin high that is hard to deny, and I shouldn't suppress. Yes! It gets sugar into the cells more than anything else, easing cellular stress. Epinephrine's crazy, absent when I'm lazy, made when I am scared, you see. Then when it flows, all my glycogen goes, and G1P grows in me. My hormones, they are treating me fine, working all of the time, don't know what I would do. Ooh! Activating protein kinase, so my phosphorylase does a glycogen chew. There is such a balance, thanks to hormones' talents, managing the energy, making glucose from my head to my toes so evenly flow in me. Thank you, guys. Whew.